coalescent theory or something, and again, you can find the title in the web page of the program. Okay, so the thing is this. So what have we done so far? So we say that I know I'm starting today is my initial condition. Okay, so I have some E. coli, which I'm putting in my petri dish today, and I want to take a prospective view of evolution. That means I want to know what will happen tomorrow, day after, etc. I've been writing equation in which I say, I give you the frequency at time t, and then I calculate at time t plus 1, right? So that's how we have been doing things most of the time. That's the classical or standard PopGen. PopGen, I mean population genetics. Since 1980s, there is another way of looking at it. Again, it's in the same field. It's still called population genetics, but it's a retrospective view of things. So everybody understand what that means? Prospective means future. Retrospective means back in the past. So the question is, today is my final condition. So what I have is my final condition, and I want to know, how did I get there? What happened in the past? What were the evolutionary forces which were shaping it, so that what I see today, how was it shaped? So how can I understand, given if I give you some samples today, what can I say what happened in the past? Okay, so I'll do very little simply because I'm with Jyoti Dalal, she's my PhD student, so we are trying to learn this ourselves. So I apologize, I'm a bit green myself in this, but I thought I wanted to share this idea with you. Okay, so here, unlike what you have been doing, this is a retrospective. Okay, so we'll have the sample today, and I want to know what happened in the past. So we want to do the same sort of stuff. And the object, what is the object that we have been concerned with in this last 3 lectures? What was the quantity we have been calculating? Allele frequency or genotype frequency here. Here, the quantity of interest is called gene genealogy. I'll explain all these terms. Okay, so I'll explain what that means. And there are some applications. So for instance, I want to know some parameters. So there are these parameters you have seen in the last 3 lectures. There was this population size, mutation probabilities, S, I didn't give it a name, but it's called selection coefficient. Okay, so these are the parameters that enter into the theory. But if I'm looking, if I have sample today, what can I say about these parameters in the past? So those kind of questions can be answered. But it requires, there is some theory, also it requires statistical analysis, which I really don't know much about. Okay, so what I'll do is, I'll set up the theory, the simplest basic one, and just analyze some mathematical results. Okay, so that's the sort of small aim I have for this part. Okay, so why should we do this? Simply because we have a lot of data. I mean, there is a lot of data now we can get, and then we want to know how can we use this. So as an application, I'll just discuss a very small one to estimate parameters. The model, and testing hypothesis about the past. So one of the most common one is to know whether neutral evolution is mostly neutral. Like let's say if I give you a sample again, today's, then what can you say whether the evolution happened neutrally or not? Was selection important? How important was that? So those are the sort of questions that people are typically interested in. But what we'll do is to start with something very simple. And we'll stick basically with simple stuff. So let me first say what this coalescent, what is this gene genealogy first? So let's go back to what we discussed yesterday, namely the Wright-Fisher process. Let's start from there. Let me give you an example here. So I'm using a little different notation. Instead of A1 and A2, I'll write zeros and ones. And I'll explain what I mean here. Okay, so these zeros here mean that I had a population at some time equal to 0. And 0 represents that all of them were same time. Okay, I should say here, today we will have no selection. We will introduce mutation. So no selection is called neutral. So this is neutral coalescent. And we will have drift. Okay, these are the elements that's going to be. So I'm going to run a Wright-Fisher simulation. I start with this population in which fitnesses are same, and all the individuals, 5 individuals are identical. That is, they don't have any mutations amongst in between. There's no difference. And I just run it. Let's say this is the result of my simulation. So remember what we said yesterday? This one chooses one of the parent. Okay, so this is fine, and this arose because of a mutation. 0 became 1 with some probability. So 1 means it acquired 1 mutation. So let me just run it for a while. So I just ran my simulation. So all I'm saying is you just throw a random number, choose a parent with a probability proportional to its fitness. Fitness, let's say it's 1 because they're all identical. Let's just set that equal to 1. So all these parents are equally likely to be chosen, 1/5 probability. And I just draw these arrows. Sorry, 2 is it acquired 2 mutations. Let me just draw this one. Let me just finish drawing and I'll explain. So this one, this produced 4 individuals, this produced 1. Okay. Okay, so I'm drawing these links because yesterday I said forget these things. Links are not important yesterday. We said we don't care who begat whom. That is, we don't care who was a parent, who was a parent of an offspring. We just cared that how many zeros and ones we had compared to what happened in the last generation when we wrote this transition matrix for the Markov chain yesterday. But today we want to focus on these links. Okay, I hope I drew all of them. So everybody has a parent, I hope. So this is, all right, yeah. Fitnesses are same. There could be different mutations. No. If there's same mutations, then it doesn't mean anything. What do you mean same mutations? So suppose, so let me just explain this. So if I say 0, I mean something like this. A sequence like this. By 1, I mean something like this. It could have also been this. And yeah, this one, the daughter cell has a 1 mutation. Right now we're not writing sequence. I'm not writing this detail of the sequence. I'm not saying where it happened. Yeah, so those are more detailed models. I'm not going to do that. Okay, we're just focusing the number of mutations, right? Okay, and similarly 2 represents something like this and other possibilities. Okay, so 2 is the mutations, 1 and etc. This means mutation, 0 means no mutation relative to some reference sequence. So this is just one Wright-Fisher we did. No, yes. So you could put 2, but this mutation probability is a very small number. Remember the numbers I said. The probability of getting 2 mutations would be mu squared. Mu is of the order of at most 10 to the power minus 4. Per base pair per generation. So I'm neglecting that, but you can put it, it's not a big deal. You could put more. Sorry? No, I'm sorry, I didn't explain this properly, I think. 2 represents something like this. 2 mutations, no matter where. 2 is a sequence which has 2 mutations. Okay? No matter where. So I'm just drawing the number of mutations, how they are changing. Is this okay? What I've drawn here? Yeah? Okay. So this is, yeah. Two parents, how can one guy have, oh, this is asexual. Asexuals. Yeah. I mean, this is all asexual, you can have diploid, etc., but let's not get into that. It just makes a difference of whether I write N or 2N in most of the cases. But this is something you have to work out and see, oh yeah, that's all the difference there is in most of the cases. Okay, so we'll just, yeah. Yeah, it can contain lots of zeros, yeah. So lots of zeros means what? Zero simply means something like, okay. So this 0 is, let's say this guy. Let's just do a little bit. Suppose 0 is this, okay, and 1 is this guy, or okay. Right now, just count the number of mutations. It's okay. Right now, I will not, for most of the time, I won't even bother about mutations. Okay, so this is fine. This is the forward time simulation, or forward time, what's going on. So one draws, a genealogy is, when you go back in time, okay, and ask, okay, I'm just going to mark the parent. This one. Okay. I think it's okay now. Okay. Right now, let's not bother about that. Nothing is entering. I'm not going to talk about that at all. We're not bothering about that. Why should it matter? But it's a per base. It's per base. This is a per base mutation. Yeah, but each base is itself small. I understand what you mean. So if L is large, yes, then there will be change. Then there's a large change. But let's not get into that. Let me just get started right now. Okay? So this blue guy, that's a genealogy. So what I have done? So what I've done is I ask, go to your parent, connect, parent, grandparent, connect. Okay? That's all. But going backward in time. That's the genealogy of the population. So the word here is genealogy. W1 of the population. So you see, they all going back and they all meet this guy. This guy, if I go in how many, 6, 7 generations, all of them had this, whatever, great grandfather to be this person. Right? So this is called the most recent common ancestor. MRCA of of the population, most recent common ancestor. Okay. So this is for the whole population. I was working with n equal to 5. So this is for population of size n. I want to introduce another number, which is a sample size, which is a smaller number, small n. So what I'm saying is, I have this large, just let me finish. I have this large population. I'm not going to collect a sample. I'm not going to look at the genome of each of us, right? I'll have this, in this billions of us, we get just something like, in Human Genome Project, there's just 1,000 genomes or so that has been analyzed, right? So that's a sample size. It's much, much smaller than the actual census or the large population size, right? So that's a little n. So suppose I was given this sample. Capital N is the total population size, which is 5 in this case. Okay? So suppose this is 3. Let's say this one, and say this. So if you don't know, the whole population is like huge. And now you just give me these 3 samples. So I can, just same way, I can just construct the sample genealogy. And this sample genealogy merges at, sorry, this is not required, over here, right? So this is the MRCA of the sample. So there are these 2 things about this, the population and the sample. Sample is much smaller. Yeah. Is it okay? Okay? Fine. So the thing here today would be to focus on these samples, which is a small number, much smaller than the total population size. Okay, so we are thinking that, we have this, somebody is giving me data of 1,000 humans, and there's billions of us, but we want to understand that. So I do not understand it because I haven't studied it enough, but I want to give you this idea about what one does with these things. Okay, so is this okay? Is everybody clear about what is genealogy? Okay, yeah? Okay, so I want to derive a famous result. It was came by Kingman, John Kingman, in '82. It's called Kingman's coalescence. So yeah, sorry, I didn't say. What is this coalescence? You see, what is this coalescence we are saying? I mean, these are coalescing, coming together. So going backward in time, the genealogies are coalescing. Coalescing is coming together. So going forward in time, things are branching out. Okay, so that's why coalescent. Okay, so let's again start with Wright-Fisher. What I want to do is, I want to start with the Wright-Fisher process. I write down a certain probability which is important, and I'll derive a result which is, sort of a universal one. And that will define my coalescent process. So I want to go through this calculation of it. So we're always going to restrict ourselves with neutral case, and right now I'm not going to even pick mutations. This is John Kingman, 1952. Okay. Yeah. You don't have to, right? Because you just stop here with a sample. Right? So you could ask the MRC of the sample, MRC of the population, but population is huge. No, it depends on the question you ask. So we will be asking questions about coalescence still here, coalescence still here. We'll see. Okay? So you don't have to there. We can stop wherever we like. No, how can it branch out if you're going backward in time? No, show me where. Where can it branch out here? So because each parent will have things, lots of links going inside it. Because, right? So each offspring has a single parent, but each parent have many offsprings. So there are lots of things going inside when we're going backward in time. That's coalescence, right? Okay, so here is the quantity And I'll go slowly that I want to calculate. Okay, so let me just define it, and when I work it out, you will see why am I been doing all this. So I want to calculate this object. Probability. So this is called a lineage. So this is one lineage. Okay, this blue thing or this pink thing. This guy's lineage is this. So the probability that I have j distinct lineages so if I had I lineages today in previous generation. So I want to focus on this quantity for some reason, and we'll see why. Okay? So what I'm saying is that I have this large population of n individuals in generation T, and in generation T plus 1, I have this little sample of 1, 2, 3, 4 guys, let's say. And so I have these 4 different ones, like here. So these are my 4 guys, but these 2 merged. So I have how many lineages? 1, 2, 3, 4. So, no, sorry, 1. So these 2 merged, so this 1, 2, 3. So this is, there were these 5 guys which are different, but these 2 found the same parent. So the number of distinct lineages has reduced by 1, right? So I'm interested in, so for example, G, I minus 1 is, if I is 4 here, the 2 of them was same, had the same parent in the previous generation. So so something like this would be captured by this probability. Okay, so I want to do this in general. Okay. So this has, so let me write it down and explain. So this is equal to number of ways So let me try to explain this by an example. Suppose I have I equal to 5, j equal to 3. So this is what I'm asking. So I'm saying I have 1, 2, 3, 4, 5. Okay, today. So this is in one generation. Yeah, in just one generation, I want to see that I have I lineages today. So, sorry, I had I today, and they reduced to j. Like j has to be less because of coalescence. So let me explain what I mean. So I'm asking, suppose I had these 5 guys in the previous generation. Something like this could happen in one generation. Any of these 2 found the same parent, had the same parent, right? Something like this could also happen. Is this clear what I'm drawing here? Right? Yeah. No, here. These 2 you're saying? This guy? Yeah, because so that's one thing. If you go backward in time, it didn't leave any offspring. So how will I, it's not leaving any trace of itself over here. We're not counting in the sense this never had an offspring. So how would I see it today? So that's why I'm saying you just, whoever is today, whoever is present, so we all have to trace our parent, grandparent, and so on. If my grandfather's brother didn't have a kid, he won't have any, I won't have a cousin there, right? So I'm saying starting with my cousins and myself and all, I just go back. So that's my genealogy. Okay, so you have to just connect, starting from, you have to work backward in time. And just keep connecting your parent, grandparent, and so on. Right? Okay, so let me try to explain this. Okay, so, yeah? Okay. So this is, now I'm saying this is a neutral case. So I said over there, offspring chooses a parent with a probability 1 by 5, right? Because it's neutral, or are equally likely to be chosen. We learned that for the right fisher process yesterday. So therefore, the probability, each of these guys chooses, so I'm not, so what I have is here, this big thing, but this dashes, I mean, I have n individuals. Okay, so these guys represent the blue guy, the pink guys. Okay, and this dot dot means I have this large population of size n. The n of these guys. So these two chose, let's say, this parent. These two chose this parent, and this guy chose this parent. Okay, so the probability of choosing a parent is 1 by n, and the I of them, I is 5 here. So that's why this factor, right? This is the standard I-Fisher pi. Let's try to understand these two other things. So this is the number of ways to occupy J out of n boxes. So what do I mean by that? So this, I'm saying, I want to get J clusters. I want to get 3 clusters now, right? So I think of it like this. So I have these n boxes, which are these, and I want to get 2 here, 2 here, 1 here. I could put anywhere. So these two, don't look at whether it's 1, 2, 3, 4, and 5, we don't care. All I care is that there is a box here, there is a box here, and there is a box here. How many are there? We'll worry about that in a minute, in each box. So I want to get 3 boxes to be occupied. So this object, when I am saying 3, these are n of them. Maybe it should be a bit more concrete. 4, 5, 6, 7, 8. I think it's 8 or something. So I want to occupy these 3 boxes here. These boxes. So what is that factor? Right? So we have to see that. So if I take this guy, this would mean I have 3 balls here, 1 ball here, 1 ball here. So I'm saying, right now, don't worry how many balls are there in each box. Right now, just say that there are 3 boxes that I want to occupy out of n of them. Doesn't matter. It's just apparent. There's nothing about, there's no space. It's just a way of drawing it. There's no real space. Okay? So I find it easier to think in terms of these boxes, but you might have a different way. So I'm saying this one also, this would be something like this, right? But don't worry whether 3 balls or how many are there. We'll worry about that in the last factor. It's just 3 boxes out of n. So what is that factor? How many ways I can do that? First box can be occupied in n ways. That means that these two could have chosen any of the n ones. It's not special. There's 1 and 2 are not any special. It's just labeling. So they could have chosen any of the n guys. I want 3 clusters. So 1 out of those n cluster, n sites has already been done. So I'm left with n minus 1. So second cluster would occupy any of the n minus 1 sites. Third cluster could occupy n minus 2, and so on. So this is for the first cluster, second cluster, third cluster. So it's better to write it like this. So if I want to occupy j of them, n is? No. I is different. I is these 5 guys. N is a very large number, say 20. These pink guys are the I's. So N is, in this case, 5. Okay? And these are the 3 lineages, the 3 guys that merged here and became, these 2 merged and became 1. So here, this lineage, this survived. And these 2 found the same parent. So therefore, the 3 became 2. Okay? Is this factor clear to everyone? This is the easy part. So this is equal to, and so is this the notation? It's called descending factor, whatever. But you know what it means. Okay, this is this factor. The last one is a little bit more involved. Let's do that. So here I'm asking number of ways of partitioning I objects into J clusters. So let me first do a simple example, and then we'll plug it back in. So this is given by what are called Stirling numbers of second kind. Okay, so what is this object? Let me just try to figure this out. Let me just do an example first. So I'm saying I have, this is I, partitioning 4 objects into 1 cluster. So how would it look? Something like this? 4 objects into 1. How many such possible ways are there? 1. This is 1. Partitioning 4 objects into 2 clusters. So I could do this, or this. I now have to put in numbers, 1, 2, 3, 4, etc. Shall we count it? So this could have been 1, 2, 3, 4. Okay. 2, 3, so 2, 3 is already done. 2, 4, 3, 4. No. 2, 3 is here. 2, 4, 3, 1, 3, 2. Did I miss 1? 3, 4 is also there. Did I miss? No, so that's right. Yeah, sorry. 3, 4, 1, 3, 2, 4, 1, 4, 2, 3. Yeah, so there are 3 ways. Sorry. Yeah, so these are 3 ways, right? 1, 2, 3, 4, 1, 3, 2, 4, 1, 4, 2, 3. Am I missing something? No, but yeah, but it's not just that, it's not like 4 choose 2 or something. Because if you do 4 choose 2, what will you get? So that took me a while. 1, 2, 3, 4 I'll write, right? So if you do that, you'll get something like this, right? It is the same. It is like saying 1 and 4 met, 2 and 3 met. 2 and 3 met, 1 and 4 met. It is the same for us. If you do 4 choose, is that what you're saying? 4 choose 2 or something like that? No, that's overcounting. No, it's not like that. Let's just do this. This is not like, I don't think you have seen, I mean, at least I had not seen this before. Let me just work it out, okay? So just stay with me and we'll see if there's any discussion. So this is what I mean by S1 contribution to S42. Okay? What about this guy? 1, 2, 3, this could be 4, and so on. So 4, 3, 2, and 1, this could be. So this is easy, this is 4. Okay, so this guy is S42 is 7. Very quickly, S43. So I have 4 guys, and I want to make into 3 clusters. And, what a permutation of this. So that's also 6. and so on. So you can work out other ones. This 4 is 1. Is this okay? Is it clear what these Stirling's number are? So this is the sort of thing that we need to count. So I said, don't worry about how many balls are you putting in each box. Let's just make sure we get 3 clusters. And now I'm worrying about which balls I'm putting in each cluster. That factor is captured by the Stirling number. Okay? So they obey the following recursion. So this is saying that I want to put I objects into J clusters is equal to, I put I minus 1 objects into J clusters first, and then I'm left with 1 object, right? But I've already created J clusters. So the leftover guy, I don't want to mess up with the number of clusters. I've already put the J clusters. The leftover guy can go to any of the J clusters. So that's why factor of J. Okay, so that's one term. This is saying I have again I minus 1 objects. I put them in J minus 1 clusters. So let's say I have 5 objects and I want to put them in 3 clusters. So I first take the first 4 objects, and the last one I just keep in a single cluster. That's why I don't have any, I just have a factor of 1. So I'll encourage you to just work it out. It's probably a new thing, but you know, just do this. It's very standard in the literature, etc. But I'm sure you can work it out. Okay? Unfortunately, we don't need so much. I'm sorry, but I thought I'll just introduce it to you. Because this is so far all exact. So what we have then is this. Minus J plus 1, yeah, and SJI. So this is the ways of, this is the probability that if I had I, they became J in the previous solution. Okay. But the way you do, you know, when you are figuring out, I think, Poisson distribution from binomials. So it's a sort of somewhat similar thing. I have I factors in the denominator. How many factors do I have in the numerator? J, right? J of them. So let me write it as It is different. No, it's not like that, right? This is just saying, so okay, let me see. If I have n equal to 4, and I want to occupy 2 boxes. So this first box can be here, second could be here. So right now, don't worry about the labels. No, 1 and 2 is same as 2, 1, 1. But I'm not worrying about the labels at the moment. So that labeling part is coming in this one. I'm saying you just put in, whether it's 1, 2, 2, 3, we don't care right now. Are you talking about this part or this part? This part, no. I just want to choose 2 boxes. I want to have exactly 2 boxes, right? So this could be occupied with any of them. So let's just work it out. So this guy could be here. How? So this is his 2, and then this is occupied. This is occupied. So this is occupied, and this is overcount. This is not correct, no? This is not correct, yeah. So I don't think I counted that. No. So this should be what? 3. 1 second, how much should be? j is 2, n is 4, and j is 2. So it's 4 into 3, n minus j is 2 plus 1, 3. 12. 12, okay. Wait a second, let me see. I think I am distinguishing between these two boxes, I think. Yeah, that's why I have this, that's why I'm not dividing it. Yeah. Yeah, there's not label, it's, I think, no, let me just think. I think I'll need to think. I'll try to clarify that. But this is correct, okay? I'll just think about it. Probably I made some assumption here. Okay, so but let's just please buy this right at the moment. I'll try to clarify later. Okay. So if I have this, then what do I get? Yeah, so these terms are, okay, so if I divide this, so I have a term of order 1 by n I minus j, right? So the term like I to I minus 1 is simply, let's do this. Is this okay? And this is saying that I have I objects and I want to make them I minus 1 clusters. That means I'm putting 2 of them in the same. That's just I choose 2. That is the simplest one. That's I choose 2 divided by n. And now if I just keep terms leading order, so this is I choose 2n, there will be terms like order 1 by n square, right? So I'm going to neglect such terms. But then if I'm neglecting all these, I to I minus 2, etc., they will be even higher order. The leading order itself is like I to I minus 2 is of order 1 by n square. Okay? So I'm going to neglect all that. So this is a scaling thing that I'm doing. I'm taking the large n limit. This is fine. This is all exact. Till here it's all fine. It's exactly correct. But now I want to work in the limit, scaling limit when n is infinity. Not scaling limit. Capital N is large. Okay? And I'm keeping only the leading order terms. I to I minus 2, etc. of higher order, I'm going to set that equal to 0. This is the surviving one. And correspondingly, for I to I, it's this. Okay? And the rest, okay? So this is, with the large, if n is large, this is the answer. Okay. Let me just do one more step and then I'll come to the main point. So therefore, so this is setting up this probability of reducing in one generation a lineage. If I now ask what's the probability that I got lineage reduced by 1 in G generations, so that's equal to, because I know this probability that how it reduces by 1 in one generation. And if I assume these are all independent random variables in the previous times, so that's simply GII minus 1 and 1 minus let me just define. So this is all discrete generations. So let me define time Just like I did yesterday, I was measuring time in units of n. Here also, if I measure time in units of n, this is a continuous time, g was a discrete one. Then when you change variables, you will get this. So the n that was here, that will get absorbed over here. Okay? So let me do this. Okay. So this is the key result. The time when measured in units of n for the Wright-Fisher process. For the coalescence event, so this is the probability that lineage decreased by 1. If I had I lineages, it decreased by 1 in time t. So as the coalescence time, when I had I distinct lineages, it reduced by 1. That probability distribution is given by this object. The important thing here is, instead of Wright-Fisher process, if I had taken other models, like Moran process, or something a little bit more complicated, but again, neutral and everything, okay? Then all you have to do is to change the scaling. So for example, for Moran process is n square. And you again wind up with this result. So in that sense, it's an important one. So this defines the coalescent process. The probability, so then what I'm going to do is to draw those kind of coalescence trees. And I want to know when, so if I now want to draw these things, probabilistically, I want to define a process. I need when the coalescence occurred. When did these two things coalesce? What's the distribution of those times? So that distribution is given by, it's an exponential distribution with a mean 1 by I choose 2. And the point is, you take other processes, you still get the same answer, provided you do the right scaling. There are some conditions, but provided you do the right scaling, you get the same answer. That's why this is the important, famous result. Okay, so I want to use this result now. Yeah. Yeah, so let me just, I should have said one point, which is, so I'm saying this I to I minus 1 when n is large. I could become I minus 2, right? But that I'm saying for large n is a next order. So when I'm neglecting these terms, I'm setting this probability. What does it mean? It's saying I'm going to consider only coalescence of 2 lineages. So something like this has a lower probability. For large n, for this neutral coalescent process, this is neglected. This is set, this probability is set to be 0 for large n. There are more important, complex problems where this is relevant. So there's a multiple mergers can happen. So this is something which we are interested in studying. But today we'll just neglect all this. So here, to arrive at this result, I'm saying I'll consider things of this kind. So at any point, only one merger happens. That's the meaning of this object. And all these rest of them are 0. So if somebody has 3 offspring, so 3 lineages are merging, those events for large n are improbable to leading orders, and those ones I'm going to neglect. Okay? Otherwise, you have the full exact result to do that. But for large n, that's the result. And I want to use that now. Okay, so let's just to understand this process a bit. Let's do a small exercise. So capital N is basically out of picture. Okay, so we have scaled the time. So whenever I say t, I just mean that I'm going to measure things in units of capital N. The thing that is going to matter is I, the number of lineages, and the sample size. Okay? So capital N is now gone. We will not worry about this large population size. Okay, so let's just do a little exercise. So allowing only 2 mergers. Suppose N is 2. I have 2 samples. I'll have, this is the coalescence tree, right? N equal to 3. I could have this 1, 2, 3. I could have this. I could have 1 and 3 merged first, and then 2, or 2 and 3 merged first. Right? There are 3 trees. I mean, so What I haven't put in is the time, that is when this happened. Or these are all random. These are all random trees, and we'll talk about it in a minute. Okay? It's just drawing these structures. What about n equal to 4? How many trees could you find? Can you draw some? So I'm not allowing something like this. This I'm not allowing. Within Kingman's coalescence process. Those things are not allowed. So we just allow at any point in time these two guys to merge. Yeah. I is the number of, because this I is going to change. Is the running variable. I start with n of them, and it's going to change. See here, I started with 3 of them. At this point, I have 2 distinct lineages. So I is now 2. What about 4? No, at not same point. This is not allowed. Yeah. In this expression, exact expression, that's allowed. Sure. But for large n, those are higher order, 1 by n square. So I'm not going to neglect that. So that's how the process is defined. Is this okay? Okay, n equal to 4. So you could have something like this. Or, okay. Something like this, but then you have to put these numbers. So first, let's say, so I shouldn't do this. 1 and 2 merged first, and then 3 or 4 later, or 1 and 2 merged, then third one, then fourth one. So this bunch, and then they're together. This bunch, and then this guy came, and this guy came along. And again, you have to put in these numbers, 1, 2, 3, 4, 1, 3, 2, 4, etc. And in this case, it turns out you have 18 such trees you can draw. So the general answer also is not difficult to write down. And I'll just tell you, in this case, 18. So what you have to do is basically say the following. So you say that, I'm just choosing any of the 2 to merge, right? So I want to go from I to I minus 1. I don't care much time. Right now, we're not bothering about that. So I can choose any 2 to merge, right? So that's, I choose 2. So I started with I of them. I chose any 2 to merge. That is, I choose 2 ways of choosing them. But then I have now I minus 1 left. I choose any 2 of them. And that's all 2. So this total number of trees in this manner is just simply this. So it's exponentially large. And you can check if we have the right answer here. So, like, if n is 10, you get about 2 million trees. Okay? So, so one has to figure out which one is right, etc. But people do some sort of statistical analysis to figure out which is the most probable tree. But I just don't know enough about that. Okay, so this was just a simple exercise. But now, this is our key result. And I want to understand this a bit. So what can I calculate? Okay, so let's start with something simple. So probability that if I have I distinct lineages, coalescence occurs in time t, and I'll write time ti. That's the result over there. Okay, so the average time is how much? Average time 1 by alpha, right? variance, of course, you can calculate. So this is the time that if I had I distinct lineages, then the time for two of them to merge is this. So let's just draw an average tree. Suppose I is 4 to start with. Total is 4, and to start with there are 4 of them. So what's the time for, so right now I'm not bothering which tree, but I'm just saying let me just draw one tree. Let's say these two merged. What's the average time? I had 4 of them, so this time is 4, 2 by 4, etc., etc. So how much is that? 1 by 6, right? So this time is 1 by 6. And these two merge, let's say this one. How much is this time? 3. So now I is 3. So that's what? 1 by 3. 2. This one. This was almost double. And sorry. Is this clear what I'm trying to draw here? So this time is T4. 4 distinct lineages I had. They became 3 at this time. So that time is T4. That's 1 by 6. And then they became 2. That time, 3 to become 2 is T3. And then this big one. We'll have lots of trees. We just drew some, right? I didn't put the times and all. But this is a typical tree in the sense. So I don't mean that, of course you could have had this, of course, you could have had that also. That's not what I mean. I mean, the time for the mergings are like this. As you go farther back, time is increasing. Does it make sense? So I'm asking, suppose in this room, we want to find our relatives, right? And whenever I find a relative, we just bunch together and then go and look for another relative. Pass, right? So initially we'll find a friend or relative quickly, right? And we'll bunch together. But as time goes, it's going to be harder and harder to find my friend or my relative in that group. So that sort of thing. So it becomes harder to find another distinct one as time goes on. Okay? So this is the property of this. So there'll be lots of tiny, tiny branches. If you look at some of the pictures of coalescent trees, you'll see that it was N10. You will see lots of tiny, this kind of sort of structure, something like this. I'm not drawing it properly, but you should look up the pictures. Something like this. And then you have something like this. The last merger takes the most of the time. And that you can see more easily if you calculate PMRCA. So it's simply the total time. Just 1 second, let me write it down. Sorry, I'm just being the same object. Yeah. Because we have to find somebody else, who is different from us. No. N is large. N is infinitely large. N is infinitely. No. I have to find. So I find my cousins, my whatever mamas and chachas and stuff. I find that, and then we go together and look for another one. See, this is the coalescence, right? These are, let's say, us, and we found our, whenever these two find out the parent, they go together, and then you keep finding grandparent, etc. It's going to be hard. So capital N is out of picture. We have to find our relative. We don't care. We don't want to find somebody else. We want to find our relative. So somebody who is, once we have all together, to find your great-great-great-grandparent, that's going to be hard. To find that lineage. Tracking that lineage is going to be hard. Capital N is large. That's, of course, but that's out of picture. That's gone. Okay, so this is this time. And this one, easy to write again. I'm just plugging this in. So if you just do this little trick, just lots of things get cancelled, and it's just this much. So the time, so this is the MRCA, right? The most recent common ancestor, where all the n of them merged. So that time is the sum of these timings. So that's equal to, so n is large enough, that's like 2 times T2. The time to the most recent common ancestor, or rather, the time for the last merger is half of the time to most recent common ancestor. That takes bulk of the time. Okay, that's the last one, is the longest one. Okay, so you can also calculate other things. Yeah. Let's see. We know that answer, right? You just tell me how much is the I. We know the time, at least for neutral coalescent. I don't know, I'm still studying that stuff. So this, see, it is of the order n. Order n. So I should just say this. So when I say n, in this business, n is not the census size. I use the word billions, but actually that's not the relevant population size. There's something else called effective population size. I haven't explained that, but that for humans is about 30,000. For Drosophila, it's more. Okay, so it's just a measure of genetic variation in a population. So the effective size of population. It's not the census, it's not a billion that matters. It's the 30,000. It works out the amount of variation that we have. According to that, you have to fit it to something else. That number is much smaller for us. So n is of the order of 30,000. So everything is in units of 30,000. Okay? Humans, it shouldn't apply because we are not neutral. I mean, there are other complications, recombination, etc. we take care of. So all that doesn't really apply. This is simply a starting point. And there are more complex models, not just the right fish, the other models that we use. The basic reason that people use this is for test neutrality. Whether the population has been evolving in a neutral, without selection or not. That's one of the things that people do. But more models that required are more involved, so I just can't do that. No, but I don't know. Do you know, Jyoti? How much is this T coalescence times work out? I don't know. But actually, that's not the thing that people actually measure. They use these fundas to calculate a diversity. This is going to be input into something else which has a biological meaning. These are okay. First of all, I think you're confusing this with phylogenetic trees. Phylogenetic trees is saying that, you know, humans and all, there was an apes, we have primates and all, if you want to go back. That's a different object. That's not what we're talking about. This is within a species, right? Yeah, so we want to know when we came out of Africa, was there a bottleneck? That means population size, did it expand? Did it reduce? How did the population size change back in time? So those kind of details you can put it in this more complicated models. So I won't be able to say more biology of this simply because, as I said already, I'm myself learning the subject. Okay, so sorry, you have to, you know, satisfied with whatever little I'm telling you today. Okay, let's just get on with this. Okay, so let's do something simple. So this is the probability distribution when I had I lineages for time ti. But you can ask for the joint distributions also. And that's simply because they don't care what happened in the past, they are just independent events. So it's simply a product. So I want to do a little thing simply because it's something you encounter in your courses as well. So this I wrote already, probability of pi. That's given to you. What about this guy? So I told you the average time to find MRCA. What about the distribution? Use the fact that the probability of T1, sorry, T2, Tn is product of those problems. what is given to you. So this what you have is, I want to sum TMRCA I've given you a distribution of each of these types, and I want to calculate TMRCA. Does this remind you of something that you've done in your very basic courses? How would you go about doing this? Huh? Right now, central theorem will come. What should I write down here? Can you say? Guys, fifth row, can you say something? Okay, so let's do it. T1, which is this? The probability that I have T1, T2. This is now just a simple probability theory problem, which is sort of very, very basic. So that's why I thought I just mentioned this. And what should I do here? Delta. And integral over with all these. Is this okay with everyone? We just want to sum the distribution of some of the random variables. It's something you learned. But I'm sure you have done this. But is there something different from what you have learned here? Is it something which strikes you is as different from what you have done in the courses? All of them are independent, that's what I've assumed, and non-identically distributed random variables. So you have here sum of independent but non-identically distributed but exponentially. No? Okay. So suppose I just wanted n equal to 2, then of course this is the answer. N equal to 3. Can you quickly try this for me, please? N equal to 3, I want to do this guy. How would you do this? So you told me the general answer, but let's see if we know how to do this. That's all I'm asking, some small thing. First integral you can do trivially, right? Zero to infinity. This is the only small tricky bit, and if you know this, then you're done. This is okay. Well, clearly not, because this is going to get cancelled, and I'll get infinity. It's wrong. I mean, what I wrote is wrong. This should be T. Why? I mean, T3 should be T minus T2, but T's are positive. That means T2 has to be less than T if it wants to click. Delta function has to click. You have to do it a little bit more carefully. And you put that limit, and then you can do for n equal to 4, 5, etc., and you'll see a pattern emerging, and then you can write down the general answer. Okay, so I'll just give you the general answer. which you can verify by induction or whatever you like. Okay. So this one I strongly recommend you to do because it's very basic. So here, this turns out to be, for this case, the distribution of MRCA. Okay, so it's complicated, but you can check it. Okay. Okay, so let me just now. Okay, so I just wanted to mention how would you. So we talked about Wright-Fisher process. Okay, so let me just finish this story a bit. So I just told you a little bit about this TMRC, but you can calculate other times, and you can build upon that to do other things, like calculating the objects that biologists are interested in. That is the measures of diversity. So I'll try to give a small example about that in a minute. But let me just say that, okay, I don't want to worry about Wright-Fisher. So imagine I have to do a Wright-Fisher process. N is large, 10 to the power 5. So I start with this full population and update each guy, make 10 to the power 5 in each generation, keep running. And am I actually interested finally in 10 of those guys as my sample size? That's clearly very inefficient, right? How would you do a coalescent process in a simulation? How would you work that? Okay, that's not difficult because now we have all the required information. So I start with, let's say, sample of 4. This is simulating coalescent. So I start with sample of 4, let's say, n equal to 4, and I just choose any of them with equal probability and merge them. The time to merge is the random variable, and I know this distribution. That's why we did all this stuff. That's that distribution. So I had 4 lineages. Probability for them to become 3 is this. So I use this distribution, draw a random number, T4, right? And put these two together. And this guy is still going. I have 3 of them now. I now change the distribution, it's non-identical distribute. I now draw random number T3, and let's say I join. I can choose any 3 of them. You'll get lots of trees, different trees, right? There are 2 types of randomness. One is which one you join, and secondly, when you join. Okay? Let's say I chose this, and then I draw another random number, and maybe this. So unlike TMRCA, unlike the average tree, I can get this tree. There's a probability for this. Right? It's just the timings being this, the product. So that's easy. It's very quick to simulate this process. So you just choose any 2, merge them by drawing a random number from that exponential distribution, which is with a I-dependent mean, and keep merging them. So this is all just drift so far. Mutations also you can put very easily, but for mutations, because you inherit it, you have to go forward in time. This, what I just described, was backward in time, but now what you do is you start from here, and then you say that, let's say the number of mutations are Poisson distributed. It's a sort of standard assumption. The Poisson distribution with some mean, mu, and then this is the length. So this is the probability per generation, per unit time, or rate. If I use the word rate, then I'm using a different symbol, u. So you have Poisson distributed mutations that happen with an average rate u. The time taken here is t1, right? Or, sorry, t2. So along this length, it could have happened anywhere. The total number of mutations that could happen on an average would be u times t2. So you draw this random number and put in some mutations. But this mutation will be inherited along all these guys. So this would inherit. Then you go to the stretch. And then again, draw a random number, and maybe another mutation occurs along this line, but this would be inherited by both the guys. then you can repeat the same thing. And maybe this guy got one more, and so on. But you have to remember to go forward in time while implementing mutations. But constructing the tree, go backward in time. So this simulation is very easy, and the key thing that we needed was this distribution. Okay. So just all you have to use. Okay. Okay, so let me just do this one little application. So suppose somebody gave me this data, and I want to calculate what is called nucleotide site diversity. So we haven't discussed this object at all in this course, but as I said in the first lecture, people worry about variation, how much genetic variation we have, how is it maintained, how is it created. So that's a sort of, one important central question of the subject. Okay, so now today's sample I want to calculate this diversity. And using this diversity, what can I say from these results? So there's one very small thing that I'll do. I'll show you only the argument, but you can actually sit down and use the result to actually work it out. Look up Blakely's book. This is also denoted by pi. Not to be confused with fixation probability, but that's, I think, relation. So this is simply asking what is the probability that two sequences differ at a given site. Now I'm going to draw something like this. Actually, before that, I think, sorry, just because I didn't demonstrate this bit, and I think it's sort of interesting to do this. It's a short exercise, let's just do this. So suppose, I'll come to this in a minute. Suppose I had this sample somebody gave me. So dot means the base pairs, the nucleotides there are same. Just like we said in the first class. We are not concerned with things that are same. We are interested in things that are different. Because that's the one important, that's where the section is going to act. Okay, so dot means these all bases are same. So I'm not going to write that. So this is another sample. These two are same, but these are not. So that's why I'm writing this base. This also happened to be this. Okay? Can you draw a tree for me? Consistent with this data. So at the end of the day, I have things like this. This is what I have today, and I want to construct a tree which is consistent with this. Neutral and mutations. Clearly they're different, so mutations must have occurred, right? So can you draw some tree? Okay, I think I should do this because you haven't encountered this before. So something like this one could draw, and you could draw others. So try doing this. This is one. So I'm saying, let's say TG is ancestor. This is questionable. But there are ways to figure out which you call ancestor. There are other quantities that one calculates, okay? So I'm not going to say what they are. But let's say TG is ancestor. I don't know who's my ancestor, to which all of them merge to. So I'm going back here. TG is an ancestor, let's say. Then if I draw something like this, G along this line became A, then this mutation happened, nothing happened here, nothing happened here. So that's how I'll get TA and TE, right? This is TG, which is this guy. Nothing happened along this line. But TG, oh, okay, T became a C here. A single mutation happened along this line. Okay? You could draw other ones. You can start with different ancestors and see what are the kind of trees you can get. So this is how you would, if this is the data, that's the sort of thing one draws. But of course, you know, as I said, even if n is 10, you have millions of trees. So you have to use some software, etc. But our interest is really in the sort of statistical properties of these. But if you are somebody who's interested in, you know, working with actual data, this is a good area. Sorry? What's that? Yeah, I assume that, yes. But it's not necessarily, you know, you could have chosen another one. And which one we choose, there are criteria, etc., that we satisfy, which I haven't talked about. Okay, so getting back. So this was one sort of simple example of if I have given sample, slightly a little bit more. So I want to calculate this object. Suppose I have some sample like this, 5 of them. Instead of T, I'm writing zeros and ones. One here. This is all same, so we should do it like this. Okay. Okay, so we want to calculate the following. So it's just a small example. I mean, it's sort of, okay, maybe I'll just give you the answer because you can just work it out. It's not no big deal. So what you have to calculate is the average pairwise Hamming distance. So this pi is something you can measure. So this sample is just given. This is something that's a thing that you calculate with your software or with your by hand or whatever. So you're just saying, I take first and second, see what's the Hamming distance. First and third, first and fourth, first and fifth. Similarly, second and third, second, fourth, second. Okay? Average over, how many pairs are there you can form? 1, 2, 1, 3, 1, 4, 1, 5, 2, 3, and so on. 10 pairs you can form. Okay? And if you count these numbers, it turns out to be, average Hamming distance turns out to be 18 by 10 pairs, 1.8. Okay, fine. And therefore, this is, if I now divide by the number of sites, that would give me the pi. Okay, but let's just keep it as 1.8. So this is something I'm just saying, this is the sort of data, the kind of data that you get and kind of things that people calculate. But this object, pi, it's saying at a site, I have one site here and one site here. I'm not bothered about right now the rest of them, just at a given site. What's the diversity? Okay. So Hamming distance here between 1 and 2 is 1. Between these two is also 1. Between these two is 2. These two is 1. Right? Then you do the same thing. This is 0, 3, 2, and so on. One, 2, 1, 3, 1, 4, 1, 5. 2, 3, 2, 4, 2, 5. 3, 3, 3, 4, 3, 5, and 4, 5. 4, 5, we're having distance is 3. Right? So that's all I've done. And the number of pairs is 10. So that's how I got 1.8. Okay? But this object you do from your data, and you can calculate it, but I'll just give you a simple argument. It's saying that I'm looking at a single site, a single change, right? So this is 0 and 1, and these are different at one site. So it could have been like this, and the mutation happened here, or 0 to 1, sorry, and this could have been like this, and the mutation happened here, with let's say equal probabilities. This time of coalescence we know, and therefore we know the rate at which mutations could have happened. That rate is 2T2, because that's the length I have along which mutations could happen, the time I had. This rate is per unit time. Total time I have is 2T2 into the mutation. So this pi turns out to be this. Average T2 is what we have been calculating, T2, T3. We said T2 is 1, but in generations it's n, or 2n depending on diploids or haploids. So let's say for haploids it's 2n. This is a simple argument, but you could sit down and actually set up a proper calculation using whatever I have said. The basic results being the distribution of TIs. So therefore, this something I can get from that sample. I just calculated 1.8 or something, right? Mu I can get, let's say, mutation accumulation experiment. I said briefly about it yesterday. And this object, n, see, I said n is out of picture. This I can get from here. The estimation of parameters is one thing that you could use, that you can use coalescent theory to do. This is very simple. This one is the simplest thing. But maybe population size was changing with time. We have to work with different models and estimate it. There could be hypothesis that, when we came out of Africa, maybe there was a lot of selection or less selection. How much was it there? Was it neutral? So those kind of things, models you can build up by going back in time, by constructing these more complicated models. Okay? But the information that you have is just this much. And you could say something about some kind of global parameter. Okay? So I think maybe I'll just stop here. But you can find more in Bagley's.